Hello and welcome everyone to the podcast for day 27. We have 3 objectives and so we'll have 3 podcasts. This is the first one. In this podcast we're going to talk about genetic variation and how that makes evolution possible. The first thing we want to talk about is phenotypic and genotypic variation. Phenotypic variation, as the name implies, is the variation you see within a population based upon the phenotypes. So let's write different phenotypes in a population. They are not heritable. Genetic variation, however, is the difference in genotypes in a population. Genetic variation is heritable. For instance, if you had a red flower, its genotype might be big R, big R. And maybe the white flower would be little r, little r. In this case, the genotypic variability, big R versus little r in the population, is directly related to the phenotypic variability. So now usually genotypic and phenotypic variation is linked. However, sometimes you can have the same genotype but different phenotypes. Your book, there are 2 examples we can talk about. The first, well, there's several examples, but let's mention a couple of them here. In figure 23.5 of your textbook, they show an example of caterpillars. These caterpillars have the same genotype at this one gene pair. Over here, the caterpillars eat oak flowers, and the other ones are raised, or they eat oak leaves. The different chemicals you find in flowers versus leaves account for a difference in phenotype between these 2 caterpillars. In one of them, they have, I'm just going to draw it as a different color. Overall, the same caterpillar, the same genotype, and they function much in the same way. They just look different because of the different kinds of food they eat, which means the different kind of chemicals that they are exposed to as they're developing. Another quick example is a particular fruit fly. They both have a genotype that is called wingless. At a lower temperature of around 25 degrees Celsius, the flies have wings. However, at a higher temperature of around 37 degrees Celsius, they have no wings. Again, the same genotype. In fact, we could give them a genotype if it's easier to understand. WW. Same genotype. Genotypic variation is the same, but the phenotypic variation is different depending on what temperature the flies are at. We see all kinds of examples of this throughout the diversity of life where the genotypic and the phenotypic variation don't match. So a question may arise as to how is genetic variability increased. This is important to consider because the genetic variability is what gets inherited from one generation to the next generation. It is what will drive evolution. The phenotypic variation is what's selected by natural selection, but what is moved forward in the evolutionary process are the genotypes. And we know for natural selection to work, we have to have a lot of variability. Well, in order for it to work well, we need a lot of variability. So how, within a population, can we increase genetic variability? There are 4 ways that we want to talk about. One is mutations. We've already talked about mutations, but we're going to talk about them in a slightly different context. The second one is gene duplications. The third is rapid reproduction. And then the fourth is sexual reproduction. Again, some of these we've already talked about, but let's look at them again and in some ways a slightly different view. Alright, so let's talk about mutations. We've already talked about mutations, so we're not going to talk about how they're formed or what kinds of mutations there are. We've already talked about neutral, silent, missense, nonsense, and all that good stuff. We've also talked a little bit about how they're caused. They've been caused during replication or during chemical exposures, some kind of environmental exposure. But what we want to ask now is, why do we maintain in our genomes mutations that are potentially or partially harmful? We might think that these mutations, which are harmful, might be removed, or have the potential to be harmful, might be removed throughout the evolutionary process. And indeed, I think if a mutation was strictly harmful and there was no way to protect the organism against that mutation, we would predict that it would be lost from the population. But we can protect ourselves against these potentially harmful mutations. So let's draw 2 chromosomes here, and on the chromosomes let's put a gene. So as the homozygous dominant, there's no mutation, so everything's fine. However, if one of them gets a mutation in, let's just give it some asterisks here to indicate there's a mutation. And let's presume that these mutations are recessive, meaning that as long as you're a heterozygote, as we see here, there's no problem. But if you see a situation where both of them are mutated in a homozygous recessive individual, it's harmful to that individual. In fact, maybe in this case that we're talking about, it's lethal. So let's just say homozygous recessives are lethal. So the question is, why would we want to keep this in our populations? Because even as the heterozygote, we know if 2 heterozygotes mate, 25% of their progeny will be homozygous recessive, which in this case is lethal. There's got to be some reason that we want to maintain this. And I shouldn't use the word want, because that implies that we're choosing to maintain them, which we're not. But why do we maintain them? We talked about one example, and that is the example of there's a heterozygous advantage. And the example we talked about was sickle cell anemia. We also know that there's examples with cystic fibrosis, where the heterozygote has an advantage against things like typhoid fever. So sometimes we maintain them because in some environments, this heterozygote has a distinct advantage. Sometimes we maintain these in our population because there's no selection against them. Now, for this to work, one would have to assume that the homozygous recessive is not lethal. So in this particular case, we would have to say is not lethal. And in fact, let's just say they're relatively healthy. So there's no reason to select against them. And so you might say, what's the reason to keep them? I can give you one example. There's several, but we'll talk about one quick example here. And that is let's draw a cell here with a receptor. And this receptor is coded for a gene called CCR5. CCR5, this gene here, will recognize other cells and antigens during the immune system response. However, if it's mutated and not present, the cells typically do okay. There's not a strong disadvantage to that. We also know that this is how HIV enters our immune cells. It binds to these receptors and then it takes the HIV virus into the cell where it causes the damage. We know if an individual does not have this CCR5 gene, and so not the protein either, that HIV can't get into the cell. People who are homozygous recessive for this mutation of the CCR5 gene are immune to HIV. And what's interesting is that this mutation, this CCR mutation, has been in our genomes, the human genomes, for many, many years. Probably close to 700, 800 years. And it originally arose in our genomes because it protected us against other viral infections. To name here real quick is smallpox, the bubonic plague. We know that these mutations protect the cells against both of these viruses. This mutation arose many hundreds of years ago, but we maintained it in the population, and we maintained it because there was no selection against it. There was no reason to get rid of it. Natural selection wasn't working against it. So we kept it. And then we discovered, quite by accident I suppose, that this mutation actually protects people against HIV. I'm going to erase this down here so we can talk about our third example. This third example, tough to come up with a word, so let's just call it for future use. I can't think of a really good term to describe that, but let me explain it because that's really the important part. So we still have our case where we have our heterozygote up here, and we can go back to being homozygous, the homozygotes being lethal, or at least very sick. But as long as they're heterozygote, they're fine. It could be that over time, say maybe greater than 1,000 generations, there's enough time for this this gene to gain additional mutations. To the point where you create an allele that is quite different than this original one. And not only is it quite different than this original one, you've generated a new function, or an altered function, let's say altered, that, I'm going to rewrite this somewhere else so we can actually read it. So 1,000 generations, mutations accumulate, and we can generate an allele that has improved function. And now, with this allele with new and improved functions, it will become selected for. So, allowing these mutations to exist as a heterozygote allows for kind of like this evolutionary experiment to occur in our genomes to try to modify these genes in such a way to generate new alleles that have an improved function. Alright, let's go to our next example of how we generate genetic diversity. And this is gene duplication. And also we're going to say something about translocation as well. So let's draw two chromosomes here. And we'll talk about this translocation real quick here. Let's say you have a gene on this chromosome here. The same gene, and it codes for some function. And let's say at this location, it's not expressed very high. So I'm going to do a dotted line, just to indicate that it's expressed, but it's not expressed at a super high level. In some cases, this gene can be cut out of here and translocated to another chromosome. Let's say it's translocated to this chromosome. And it happens to place itself in front of a promoter that is much stronger, meaning it's going to express this at a higher level. By doing this, we've created additional genetic diversity, or rather I should say additional genetic variation. Organisms that have a more expressed gene compared to a less expressed gene will have, within a population, will increase the variation at the genetic level within that population. So again, moving a gene to a new location increases genetic variation. Now let's talk about gene duplication. We can use a couple examples. Your book uses one, and I'll start with that one. So you have a gene here, and let's use, like I said, your book describes this one with an odor detecting genes. We suspect that an ancestral mammal, so let's write ancestral mammal, small mammal, had one of these genes that was an odor detecting gene. Throughout the course of evolution, this gene has been duplicated many times. Sometimes they're right next to each other, but they don't have to be. They could be duplicated and placed on a different chromosome entirely. To the point where we know in humans we have about approximately 350 odor detecting genes. And so this is humans. And we know mice, they have approximately 1,000 odor detecting genes. They gain these new genes by duplicating them. And then, since you have multiple copies of these genes, you can gain mutations on them. And different mutations will lead to different alleles, each with a different odor detection ability, expressed as the phenotype. And you can imagine how this could be advantageous to an organism within a population in their ability to detect various kinds of odors for survival. And since we're talking about it, I want to tell you about one here that's very specific. There's a gene, and we still have it in our genome today, called myoglobin. You don't have to know too much about it, but you should know that its main job is to bind oxygen and deliver it to cells. We know that this myoglobin gene experienced a gene duplication. Now, you only need one myoglobin to survive. Well, I should say we don't. We need something else. But the ancestral organism only needed one of these. When we duplicated it, shown here, it could now accumulate mutations. As it accumulated mutations over many generations, this duplicated myoglobin gene gained a new function and became hemoglobin. It could do this because it had the myoglobin here. Without the duplicated copy, for instance, if we took on mutations in this gene here, it would end up being lethal for the individual. So that's not good. But as long as you duplicated it, you can do whatever you want to this gene here, this duplicated myoglobin gene, and you're going to still be okay. Because the myoglobin gene is still intact and still doing the job. But what happened here is, over time, we developed hemoglobin. Hemoglobin is a little bit better than myoglobin because it can bind 4 times as much oxygen as myoglobin. An organism that can bind more oxygen and deliver more oxygen to developing tissues allows those organs in that organism to develop more features and to develop better over time. So this is a good thing and has been very useful in our development. And then on top of that, if that wasn't enough, this hemoglobin now duplicated a few more times, but let's just talk about these two. And then over time, it gained new mutations, these new ones here, to give us fetal. Let's write it in blue to be consistent. Gave us fetal hemoglobin. This was adult here. And this other copy here, again, took on mutations and it functions as an embryonic hemoglobin. And as you move on in the biology curriculum or in some other curriculum, as long as you stay within the health sciences, you'll learn more and more about how it was really important for, or how it is important for humans to have these three kinds of hemoglobin, which has allowed us to develop into what we are today. And it was all because of duplicating that gene and allowing that duplicated gene to be sort of experimented on while the original one kept doing the job. So, gene duplications are incredibly important in increasing genetic variation within a population. The next two are not going to take quite as long to explain. And so the third way that genetic variation is increased is with rapid reproduction. So we can compare a few different organisms. We know that humans, we don't have a rapid reproduction. And because of that, we don't increase our variation as fast in fact, let's put humans here, compared to, say, a bacterium. Well, bacteria, we would use plural. Because these divide every 20 minutes. They have a much more rapid reproduction. And because of that, within a population, you will have greater diversity of the genomes in comparison to humans. I mean, we're more complex, but the variation that bacteria have within any given population at their different alleles is much greater, in part because of this rapid reproduction. You could take an extreme here and think about something like an elephant or a gray whale, which have even a slower reproduction rate than humans. A good example, we've already talked about HIV, but HIV has an incredibly fast generation time. It uses the human cells in order to divide, so it's not quite as fast as bacteria, but it's still pretty fast. So they divide or replicate about once every 2 days. And because of that, what happens is they gain a lot of mutations. And this has really hindered treatment. For instance, if you have a bacterial infection, you'll go to the doctor and the doctor will prescribe you an antibiotic. And that antibiotic goes after that bacterium in one way. But HIV you can't do that with, because it divides so fast, it gains so many mutations because of this rapid reproduction, that it becomes immune to those single drugs. So with HIV, you've probably heard this term before, but the treatment consists of a cocktail. Well, what they call it, cocktail. Now, this is multiple drugs in one dose. The treatments attack HIV in multiple different directions, and in doing so, it's more difficult for HIV to develop a resistance to just one method. Okay, let's talk about the last example of ways that we increase genetic variation in a population. And this should be review, and that is just by sexual reproduction. And if you remember, within sexual reproduction, there are 3 different ways that we can increase genetic variation. The first is crossing over. If you remember, during prophase 1 of meiosis, crossing over occurs between homologous chromosomes, and that increases genetic variation. The next way is independent assortment. Sorry about that. And remember, this has to do with the way the chromosomes line up during metaphase 1. And then finally, fertilization, random fertilization. All 3 of these, which we have already learned about, increase genetic variation within a population. Okay, that ends the podcast for Objective 1. If you have any questions, please make sure you come see us and we'll answer any questions you may have. We'll see you in class. Bye.